All right, so we are on chapter 6, and we're going to talk about protein function. This is a really, really cool chapter because proteins are responsible for so many different cellular functions. And so in this chapter, in the very beginning, we're going to talk just about the major functional classes of proteins. And then we're going to cover how protein structure mediates protein function. And we're going to do that by looking at, in the remaining 3 sections, the 3 different examples of that structure function relationship that we've kind of been talking about for I don't know how long, the whole the whole semester so far. So there are 2 main concepts for this chapter, really. And the first one is that all of our protein functions are really due to precise structural interactions within proteins, so within one individual protein and between protein to protein. So it's all about precise structural interactions. And we're going to see that illustrated in each of our examples. And the other one is that the binding of really small molecules, ligands, right, to a protein can induce conformational changes, right? And so if our protein function is due to structural interactions, right, then if we induce changes in the conformation, that's going to enable our protein to perform its function. And so that's where we see that really, really nice relationship between structure and function. So let's start out with our 5 major classes. They are proteins that are involved in metabolism, structure, transport, cell signaling, and genomic caretaker. I've never heard it called a caretaker before. That's kind of cool. I like that they do it that way. So some proteins that we're going to talk about don't quite fit in any of these categories. There are some exceptions, and some will fit in more than one of these functional classes. But these are the most abundant classes within the cell. So let's look at kind of some examples of each of these. Okay, so metabolism, enzymes that are involved in metabolism. These are enzymes that are going to function as reaction catalysts. So they're going to control a metabolic flux inside of the cell, right? They're going to accelerate things, or they're going to help reactions to go. Then you're going to have structural proteins. Structural proteins all maintain the integrity of our cell. So like the cytoskeleton here, and they also help to promote changes in our cell shape. So they kind of have integrity and changes kind of going for them. Then you have to think about transport proteins. So transport proteins are going to be those ones that facilitate the movement of molecules either within the cell or between cells. So we're kind of only seeing here transport proteins, right, that are either taking molecules in or pushing molecules out. But we do have flux within our cells, and we'll talk a little bit about that. Let's see what else we got. We have cell signaling. So our cell signaling proteins, right? These function to transmit extracellular and intracellular signals. So sometimes you're going to pick up a signal from the outside, and then you're going to try to bring it in. And then you also have to have signals within one cell, right, to facilitate interaction of all the parts of the cell. So you'll see them functioning as molecular switches. A lot of times people call them that, a molecular switch. Depending on the conditions, you can turn things on or off. We'll talk in lots more detail about that. Then we have our genomic caretaker proteins. Our genomic caretaker proteins have kind of a twofold job. They need to take care of the integrity of the genome, but they also need to make sure that genomic information is accessible so that the cell can do what it needs to do and get that information. All right, so we're going to start talking about our metabolic enzymes. And I'll give you a little caveat, right? Our metabolic enzymes are usually proteins, but there are examples of RNA being an enzyme. So just remember that that's a possibility, but what we're really going to talk about are the proteins. So these are reaction catalysts, right, that can control our metabolic flux, but they are going to provide an immediate environment for that rapid conversion of our reactants to our products to catalyze our biochemical reactions, right? So remember that enzymes are not consumed in chemical reactions, right? They can keep working over and over and over and over again. And how do they do it? They do it by lowering the activation energy of our reactions. And chapter 7 is going to be all about enzyme mechanisms. And so we'll go into a lot more detail. But they're going to increase the rate of product formation because they've lowered that activation energy. And so you have to remember that enzymes are not going to alter our equilibrium concentration of our reactants and products. All they do is lower the activation energy. And this is true for their activity both in synthesis of macromolecules and in degradation. So what you're going to see is inside of any kind of an enzyme, it's the amino acid side chains that actually specify the shape and the chemical environment of the enzyme's active site. So amino acid side chains determine the shape and chemical environment of the enzyme's active site. Remember, the active site is where those reactants will bind to the enzyme. And so this is what's allowing for the lowering of the activation energy of the reaction, right? And enzymes are insanely efficient, right? They've been shown to increase the rate of reactions by up to 100,000-fold. That's awesome. So just kind of keep in mind that enzymes aren't usually all by themselves. They're usually components of big multi-subunit complexes, right? So you can have anywhere from like 2 to 60 homo or heterogeneous protein subunits. So just make sure you kind of keep in mind that it's not all by itself. Usually it's kind of in a complex, but when we write them and we talk about them individually. So it's really nice that when we talk about examples of enzymes, that usually we name the enzymes based on the name of one of the reaction components and a description of the reaction mechanism. So here's an example, malate dehydrogenase. Ooh, okay. What is it going to do? Well, it's going to catalyze a reaction that's going to oxidize, or what's another word for oxidize, right? That's dehydrogenate, right? So this is another word, oxidize. Citric acid intermediate, and the citric acid intermediate is malate. So the reaction is going to form oxaloacetate, and it's going to use the coenzyme NAD, right? Nicotinamide adenine dinucleotide. Woo! So NAD+, right? That's our oxidized form, right? And we're going to talk about this, and we're going to talk about some other enzymes in this chapter, but this is just an example so that you can see kind of how they're named. And we know, right, if it's an enzyme, it's going to end in the ASE, so that you can kind of start to get an idea just looking at the name of what that enzyme is going to participate in and what it's going to do, right? Okay, so let's move on to another example. We're going to go through structural proteins. Structural proteins are really the most abundant proteins in living organisms. There's a ton of them. And they maintain cell shapes. They promote changes in the cell shape. So think about cells that have to actually move. They have to change their shape in order to get from place A to place B. So they're going to be very dependent upon those structural proteins. They can also serve as a framework. So an internal skeleton for individual cells, for tissues, for organs, right? And so a subset of these structural proteins, for example, are cytoskeletal proteins. My favorite proteins ever. So awesome. All right, so we talked about cell shape, we talked about cell migration, and they're also involved in cell signaling, which we didn't say yet, but they are. So some examples are actin and tubulin. These two, actin and tubulin together, are the most abundant in animal cells. And the two of those will form really long polymers. So you got like monomer after monomer after monomer that assembles into a polymer. And the polymers have hundreds and hundreds and hundreds of monomers, and they self-assemble. So it does not require another protein or enzyme to catalyze that polymerization. It does it itself. And then we also have collagen. Collagen's a major structural protein in animals. So let's look at these examples. This is my favorite protein on the planet. Actin. It is a structural protein. So this one we find in animal cells, and we have different forms of actin inside the cell. And one of those forms is found in muscle tissue and is responsible for the scaffolding for the contraction of muscles. And so we said that they self-assemble, right? And they form polymers. And once we form those polymers, we now call them by a different name, and we call them thin filaments. And so, right, our muscle tissue really consists almost entirely of structural proteins that function like a molecular cable, and they're going to mediate movement through an ion, through calcium stimulated muscle contraction. And so really you have actin as like the scaffold, and then you have myosin as the motor that's going to make that contraction happen. And so we'll talk a lot more about that later. But what you can see here in our diagram are that here are some individual, here's a monomer, here's a monomer, here's a monomer, here's a monomer. So these monomers are assembling in order to form a really long polymer. And so if you were to look at a cell and specifically look at the actin, what you can see are actin thin filaments that form a network inside of the cell. So you can see them running through the cell in different directions, right, to control that cell shape. And then, do you see these protrusions out here? This is the cell moving in this direction, putting those little filopodia out and just kind of going in the direction they want to go in. And so that's dependent upon on actin assembly. All right, so then if we look at our other structural protein, tubulin, and we're going to kind of compare tubulin and actin. And so these are two pictures of the same cell, but we're looking at, in one case, actin, and in the other case, microtubules. And you can see that they don't really overlap, that they actually have different functions. So they're still structural, but we have different things going on. Right? It's very, very abundant. Tubulin is very abundant like actin. And it also self-assembles. But in this case, when we self-assemble our tubulin monomers to form a polymer, now we're going to call them microtubules. And so the microtubules really are like intracellular roads. And motor proteins can hook onto organelles and actually like tow them and then walk along this microtubule road so that we can move organelles. It's also true for movement of chromosomes during cell division. It's a lot of times what we see in all those pictures that we've seen of cell division. You're looking at microtubules. But both of them are extremely, extremely important in animal cellular structure. So the last cellular structural protein on the list was collagen. Collagen is a major structural protein in animals, but in specific places. So all connective tissue is pretty much mostly collagen. And the collagen is included in other tissues like tendons, cartilage, bone, teeth, and collagen gives those structures strength, so that they can withstand a lot of stress and torsional strain and things like that. Our big classification, this is, I think, our second one. So we have transport proteins. Transport proteins are really there to facilitate movement. So that's movement within a cell, within one cell, or movement from one cell to another cell. Could be really close, could be really far away. And so you'll find them a lot of times in the plasma membrane. And when they're embedded in the plasma membrane, really what you want them there is to facilitate movement of polar or charged molecules into and out of the cell. So we have two major classifications. We have passive transport proteins. So these function in response to chemical gradients. So this is chemical gradients or diffusion, right? So passive does not require energy. It doesn't have to put anything in to make it happen. The opposite of that are our active transport proteins. These do require energy, and they're usually from ATP or an ionic gradient. And when they get that energy, usually there's a conformational change that's induced that opens or closes a gated channel to allow whatever it is to pass through. So let's look at some more examples. Passive transport, right? These are energy independent, don't have to put anything in. And it allows our molecules to move across in response to chemical gradients. So let's talk about a few of these. Porins. Porins allow small molecules, for example, water, glycerol, things like that to pass through the membrane. Now, ion channels. Ion channels are a little bit different. Ion channels are responsible for diffusion of charged ions, right? Ions, right? So hydrogen, potassium, sodium, all these are so important in cell signaling. Calcium, right? All of that stuff across the membrane. So I'll give you one example. There's a calcium ATPase transporter protein. And this protein pumps calcium across the membrane to maintain our calcium concentrations. But what it can do is when it's active, it can change our calcium concentration such that it is 10,000 times higher on one side of the membrane than the other side of the membrane. That's a lot. And it's really, really important for this, this protein's function, right? So here's a picture of this calcium pump, and it's super important in cardiac or heart muscle tissue, right? So every time our brain sends a signal to our heart in order to pump, right, it's going to lead to a very, very rapid increase in cytosolic calcium. And so you really have an increase in cytosolic calcium of like 100-fold. And so that increase in cytosolic calcium is going to stimulate muscle contraction. So the heart knows to start beating. Then the contraction is going to be over, and you don't want that calcium still sitting around. So the calcium ATPase transporter is going to pump that cytosolic calcium back into an intracellular compartment. That intracellular compartment is the sarcoplasmic reticulum, and we'll talk about that later. And that's really important because you need that heart muscle to relax so that it can then contract again. And all of that happens in less than a minute. In less than a second, in less than a minute, in less than a second. I was like, what, a second? Because your heartbeats are so rapid. So this stuff has to happen on an even faster scale. So you have to be dependent on ATP energy in order to make that happen. Otherwise, you can't make that happen quick enough. All right, so let's talk about another transport protein. This one's a little bit different. This is a soluble transport protein, albumin. Soluble transport proteins are a little less abundant, but they're very important, right? You can have proteins that transport oxygen, like myoglobin and hemoglobin, and we'll talk about hemoglobin in section 2. And so the albumin transports lipids, right? Hydrophobic molecules, lipids, right? So we actually used this protein in lab, we use BSA, right? So this is bovine serum albumin. So it's found in the serum of the blood, and it transports fatty acids throughout the circulatory system, right? So our blood is an aqueous environment, and so it can transport through there, or you can have transport proteins that function just within the cell. And, you know, the intracellular environment is also aqueous. So either way, you're going to need some kind of a soluble transport protein. So whenever these transport proteins bind to their ligand, like when albumin binds fatty acid, right? The ligand binding, excuse me, is going to induce that conformational change in the protein. And so that ligand binding is key to our protein's function, because once that ligand binds, it induces a conformational change, and now that protein can do its function, right? So let's see. The next ones are cell signaling proteins. All right, so these are, I think, number 3, cell signaling proteins. So this is actually a big area of active research right now. There's a lot we know, but there's a lot we don't understand about cell signaling proteins. And so there are lots of people studying it right now. So this is our kind of molecular switches that we were talking about earlier, right? We can turn things on, we can turn things off. And so you can have receptors that are embedded in the membrane. So we have 3 different kinds: G protein coupled receptors, receptor tyrosine kinases. So you know what tyrosine is, and you have an idea of what a kinase is, right? We're talking about phosphoryl groups. And then you can have growth hormone receptors, which are super important. We'll talk a little bit about those. You can have nuclear receptors, right? Because sometimes you need to get a signal into the nucleus to affect transcription, translation, right? Then you have intracellular signaling proteins. So you might receive a signal on the outside of the cell, and we need to propagate it in the inside of the cell. So we have kinases and phosphatases to add and remove phosphate groups. We have intermolecular adapter proteins, and we have site-specific proteases. So those are just kind of some examples of the different kinds. So I'm going to talk a little bit in the beginning here about our membrane-bound receptors. Whoa. Okay. So our membrane-bound receptors include 3 main types. We have G protein coupled receptors. So these are androgenic receptors. So those that bind to like epinephrine and things like that, those kinds of ligands. We have receptor tyrosine kinases. So ones that are responsive to things like insulin, right? So they're gonna phosphorylate on kinase residues. Then you have growth hormone receptors. These I'm going to show you a picture of, right? But once they bind to their ligands, they're actually going to, excuse me, they're going to form dimers inside the membrane. And once they form dimers, then they can now propagate or transduce information throughout the cell. So here's an example. This is erythropoietin. I don't know if y'all have heard this before, EPO. So this is the receptor for EPO. So in yellow here, this one right here, this is the hormone. So this hormone stimulates red blood cell production. And this is the hormone that was a big deal a few years back, probably more than a few. You remember Lance Armstrong when they had all the deal about blood doping and things like that. So that was all about one of the ways that you can blood dope is to take an artificial hormone that stimulates what erythropoietin naturally does to increase your red blood cell count. And if you increase your red blood cell count, now you can go farther with less oxygen, right? Because you have a higher red blood cell count. So anyway, sidetrack. All right, so erythropoietin. So the hormone binds, right, to our erythropoietin receptor. And so you have 2 subunits of this receptor, so 1 and 2. And they're gonna bind to the erythropoietin hormone. And when that happens, the rest of the hormone is going to dimerize. So it's gonna have a conformational change. And in this case, the conformational change dimerize. is that it will dimerize. Not every conformational change is the dimer, but in this case it is. And so once that in the cytoplasmic tail of that receptor dimerizes, right, it's going to stimulate the formation of an intracellular protein kinase complex, right, in the cell. So you have protein kinase complex, right, that's responsive to that dimerization. So once that dimerization occurs, then you're going to propagate this hormone growth signal throughout the cell, right? Because you get signals outside, but you need to make an effect eventually, usually in the nucleus. And so we need some way to propagate that signal from outside to inside. So that's how we do it. The receptor on the outside will undergo a conformational change, which will then signal the next thing and signal the next thing. So we'll see that over and over again. All right, nuclear receptors. So you have to think, if you're going to propagate a signal and you want to get to a point where you're going to change the environment of the cell, you're going to have to get that message to the nucleus to regulate gene expression, right? And we'll talk in a lot more detail about this, I think it's chapter 8. But you have to think of some examples, right, where you have maybe a hormone going through the bloodstream and you want it to change transcription of genes. So an example of that is estrogen and progesterone, right? These steroid receptors are major targets for the pharmaceutical industry, right? So these are the hormones that are in birth control pills, right? So you have to understand how those interactions work in order to regulate nuclear responses. And so how do we do that? We do that through intracellular signaling proteins, right? So here are our molecular switches. When we bind to a ligand or something like that, right, it's going to activate our receptor. Once our receptor is activated, that's due to a conformational change in the protein. So some examples of these kinds of intracellular signaling proteins are things like adenylate cyclase and all kinds of different protein kinases. So these signaling proteins are going to directly activate protein targets. So they go in and, for example, the protein kinases will put a phosphate on their protein target. And when their protein target is phosphorylated, all of a sudden it's active and it can do its job, right? So you got to think about these things as kind of a cascading system. So if it's a cascading system, you don't want to be permanently stuck in that signal. So a lot of times what these are reversible protein phosphorylations. So you need to be able to stick the phosphate group on, and you also need to be able to easily pull it off. And so that's why we say it's reversible. So we're usually phosphorylating serine, threonine. There's also tyrosine, excuse me, phosphorylations. And so if you target those particular proteins, right, that have serine, threonine, and tyrosine residues. Why are you doing that? Well, you had this upstream receptor activation signal, which then activated our molecular signaling proteins, right? Our molecular switches, which then phosphorylated or dephosphorylated a target. So some examples of these are our myogen activated protein kinases. They're MAP kinases. Protein kinase A, pKa, both of those two are going to phosphorylate serine and threonine. The Src kinase, these actually phosphorylate the tyrosine residues. And then phosphoinositase 3 kinase. This one is activated actually by insulin receptor signaling. So this is insulin receptor signaling. And it phosphorylates actually on the carbohydrate functional group of our phosphatidyl inositol. And so it's a phospholipid that's in some membranes. And so you just got to kind of remember, it's not always going to be serine, threonine, tyrosine phosphorylation. There are some examples where there are other things that are phosphorylated. All right, I think we're on group number 4, right? Group number 4, we're on genomic caretaker proteins, right? Now, when you think about genome caretaker, what do you need to do? There are 2 big functions that you need to do. You need to maintain the integrity of the genome. That genome has to be the same for the entire life of the cell. So you don't want changes to happen, mutations, things like that, right? But at the same time, you need to be able to have access to the genomic information, but you don't want access to everything. You want access to specific genes for a specific cell type, and you want to be able to have access to specific genes in response to the changing biochemical needs of the cell or of the organism, right? So you have to take into account both of these 2 functions of genomic caretaker proteins. They're really, really important in repairing mutations, because mutations happen all the time. And it's not such a big deal in cell types that aren't passed on to offspring, but in cell types that are, like in the germ cells, you want that fixed before they're inherited by the offspring. So you're going to have proteins that are involved in DNA replication, DNA repair, DNA recombination, right? DNA polymerase, we actually have 5 types of DNA polymerase in prokaryotes and eukaryotes. You have DNA ligase, right, to repair those breaks. You have single-stranded DNA binding proteins. I don't know why your book didn't put those in, right? So that keep that single-stranded DNA apart so that you can do these different functions of repair, of recombination, of replication. You have topoisomerases. Topoisomerases to reduce that, that strain on the DNA when you unwind it. You have DNA primases, so to prime the area for replication, right, to get it ready. You also have photolyase. Photolyase is an example of repair. This repairs our thymine dimers, right? You know, if you go out and you get UV radiation, you are actually creating mutations inside of your DNA, thymine dimers. So please, please, please don't go to a tanning booth. All you're doing is making thymine dimers. You also have caretaker proteins that are involved in gene expression, right? So RNA polymerase. So when you think about this, you need to think about, what does the cell in general need to repair damage, right? How does it know how to replace the damage? Well, a lot of times what the cell does is it uses DNA recombination. And so you have to have a plethora of proteins that allow that process to happen. And so a lot of times what we do is we look at simple organisms in order to figure out how things work. And so we looked at one of a caretaker protein in E. coli. And well, it's actually 2. It's actually the Rec-A and Rec-BCD proteins. So these are very well characterized in E. coli, right? And so there's a heterotrimer. So this is a heterotrimer, meaning that we have 1, 2, 3 different pieces, the BCD subunits, that come together. And so this complex, these parts assemble into this complex. They bind to double-stranded DNA. So you see this is double-stranded, right? And so what it actually functions to do is to unwind the DNA. And so we unwind the DNA and we start to end up with single-stranded pieces. And it helps to do this with an ATP-dependent helicase function, right? So this is actually helping to separate the DNA. So it's a helicase. And so now we have DNA sites, single-stranded DNA sites that are exposed. And so then the single-stranded DNA binding protein, Rec-A, can come in to these sites. So Rec-A is our second protein. And this is actually going to mediate DNA repair and recombination. DNA repair and recombination. So it's not usually just one protein that's required in order to do a function. Usually it's kind of a collaboration between a couple of different proteins. Okay, so once there's DNA repair and things like that, but there's also gene expression. How do we get access to those genes that we want for this particular cell type? And so there are proteins that regulate gene-specific RNA synthesis. And whenever we say RNA synthesis, what do we really mean? We mean gene expression. So there are 3 primary RNA polymerases in eukaryotes, and each of them has a very specific function. So RNA polymerase 1 is going to transcribe our ribosomal RNA genes to go make those ribosomes, right? To go make those proteins. RNA polymerase 2, on the other hand, specifically transcribes protein coding genes. So genes that will turn into actual proteins. Then you have RNA polymerase 3, which is going to synthesize all of our really small, weird RNA molecules like microRNAs and things like that. So our RNA polymerase activity of any of these 3 is going to be dependent on transcription factors that are assembled onto the DNA. And so those transcription factors can promote RNA activity, or they can inhibit, and I guess I should have said here, RNA polymerase activity, right? So transcription factors can make it easier for the RNA polymerase to work, or it can prevent RNA polymerase from actually accessing the gene. And you want that regulation to be able to go both ways so that you can turn things off when you need to turn off, and you can activate when you want to activate. All right, that is the end of section 1. Not too bad.